The nature of light. We, along with plants and animals, are alive because of photosynthesis releasing oxygen into the air. In order for photosynthesis to occur, light energy from the sun must be captured. Light is the form that energy from the sun takes when it travels to Earth. Sunlight, which we perceive as white light, is a mixture of different wavelengths. Our eyes are able to see the different wavelengths of the visible spectrum in different colors. Pigments are defined as light-absorbing molecules which plants use to gather the sun's energy, and chlorophyll is the plant's principal pigment. There are two types of chlorophyll found in plants, chlorophyll A and chlorophyll B. Both these absorb light very well in the blue, violet, and red regions of the visible spectrum, but do not absorb light well in the green region, instead reflecting green light. This is the reason that most plants have a green coloring. Plants contain red and orange pigments, such as carotene, that absorb light in other regions of the spectrum. Most of the time, the green color of chlorophyll overwhelms the accessory pigments, so we don't even notice them. But as the temperature drops in the fall, chlorophyll molecules break down first, leaving the red and orange pigments. This is the cause of the red and orange coloring of leaves in the fall. Because light is a form of energy, any compound that absorbs light also absorbs energy. Chlorophyll absorbs visible light especially well. When chlorophyll absorbs light, a large fraction of that light energy is transferred directly to the electrons in the chlorophyll molecule itself. By raising the energy levels of these electrons, light energy can produce a steady supply of high energy electrons, which is what makes photosynthesis work. Photosynthesis takes place in organelles called chloroplasts. The chloroplast is a double-membraned organelle found in plant cells and some algae. It consists of the outer membrane, the smooth outer layer that separates the chloroplast from the cytoplasm, the inner membrane, a second inner layer that contains transport proteins and helps maintain the chloroplast shape, the stroma, the dense fluid inside the inner membrane which contains enzymes, starch granules, and DNA. It's where the Calvin cycle, or light-independent reactions, which are a part of photosynthesis, occur. The thylakoids, membrane-bound sacs inside the stroma, stacked into structures called grana. These contain chlorophyll, the pigment responsible for light absorption during photosynthesis. The granum, plural grana, stacks of thylakoids where the light-dependent reactions of photosynthesis take place. This structure allows the chloroplast to efficiently capture light energy and convert it into chemical energy for the plant. Moving on, here's a diagram of the light-dependent reactions. The reactants of light-dependent reactions are light energy and H2O, or water. The products are ATP, NADPH, and O2, or oxygen gas. In light-dependent reactions, light goes into photosystem 2 and photosystem 1. At the same time, H2O goes in. The light excites the electrons inside the thylakoid, where it takes place, and the electrons are transferred into an electron carrier, initiating the electron chain. The energy from the light splits H2O into O2 and 2 hydrogen ions, which releases oxygen. The 2 hydrogen ions replace the ones lost in the electron transport chain by the proteins in the chain to pump hydrogen ions from the stroma into the thylakoid space. They pass to photosystem 1, and the hydrogen ions recharge from the sunlight. NADP+ in the stroma picks up the hydrogen ions, which turns it into NADPH. The leftover hydrogen ions pass through ATP synthase and make it rotate. As it rotates, it binds ADP with another phosphate group to produce ATP. Here's a diagram of the Calvin cycle, also known as light-independent reactions. In the Calvin cycle, the reactants are carbon dioxide, or CO2, ATP, and NADPH. The products are G3P, RUBP, ADP, and NADP+. The Calvin cycle builds stable, high-energy carbohydrate compounds by using ATP and NADPH from the light-dependent reactions. Occurring in the stroma, it starts when CO2 enters the cell. The CO2 combines with 5 carbon molecules to form 3 carbon compounds. Next, ATP and NADPH convert 3 carbon compounds into higher energy molecules. To produce the glucose, the 3 carbon molecules exit the cycle in order to form glucose. Lastly, the 3 carbon molecules are recycled into 5 carbon molecules again, and the cycle repeats. So just to summarize, the light-dependent reactions in Calvin cycle are 2 key stages of photosynthesis in plants. The light-dependent reactions occur in the thylakoid membranes of the chloroplast, and light energy is absorbed by chlorophyll and other pigments, which excites electrons. These high-energy electrons are transferred through the electron transport train, producing ATP and NADPH. Water molecules are split to release oxygen as a byproduct. Then the Calvin cycle, occurring in the stroma of the chloroplast and does not require light. ATP and NADPH produced in the light-dependent reactions are used to convert carbon dioxide into glucose through a series of enzyme-driven steps. The Calvin cycle involves carbon fixation, reduction, and regeneration of RUBP, a molecule needed to continue the cycle. Together, these processes enable plants to capture light energy and convert it into chemical energy stored in glucose, which provides food for the plant, so then when it goes through cellular respiration, it can have energy. Lastly, there are 3 different types of plants. C3 are the regular plants. C4 and CAM plants are mostly built for more of a hot and dry climate. So CAM and C4 metabolism are 2 types of photosynthetic pathways that help plants conserve water in hot and dry environments. CAM metabolism in CAM plants like cacti and succulents fix CO2 at night when temperatures are cooler and humidity is higher, storing it as malate in vacuoles. During the day, they close their stomata to minimize water loss and convert stored malate back into CO2 for use in the Calvin cycle. CAM is a temporal separation of CO2 fixation and the Calvin cycle. C4 metabolism. C4 plants such as corn and sugarcane fix CO2 and mesophyll cells into a 4-carbon compound, which is then transported to bundle sheath cells, where the CO2 is released for the Calvin cycle. This process minimizes photorespiration by concentrating CO2 around the enzyme rubisco in the bundle sheath cells. C4 is a spatial separation of CO2 fixation and the Calvin cycle. Differences from C3 plants. C3 plants, like most trees and crops, fix CO2 directly into a 3-carbon compound via the Calvin cycle. C3 plants are more prone to photorespiration, especially under high temperatures, as rubisco also fixes oxygen instead of CO2. CAM and C4 plants have evolved specialized mechanisms to concentrate CO2 and reduce photorespiration, making them more efficient in hot and dry environments than C3 plants. In summary, CAM and C4 plants have developed different strategies to conserve water and reduce photorespiration, with CAM focusing on temporal separation and C4 on spatial separation of CO2 fixation.